to this video. In this video we are going to see about fungi in their systemic mycosis, and this is part 2. This is a continuous video. In previous part is in fungi systemic mycosis part 1, and just let's move on to this video. So we already saw the general introduction of fungi. So fungi being an unique organism, it's a eukaryotic non-motile and possesses certain distinct features. And so here in this video we mainly focused on medically important fungi. Although there is a vast diversity in the kingdom of fungi, only 100 species are of medical importance and causes various human infections that are usually termed as mycosis. Such infections are of 3 clinical types. So first one is superficial mycosis, which cause infection on top layer, on hair, skin as well as nail. And subcutaneous mycosis cause infection on the surface as well as subsurface layers, and then systemic mycosis. Here in this type of mycosis, the fungi infection spread throughout the body, which is termed as disseminated disease. Well, here we mainly focused on systemic mycosis. In that we are going to see about histoplasmosis. It's an example for this deep mycosis. Well, and histoplasmosis, so it's a disease caused by means of fungi. Okay, and transmission mainly done by means of inhalation of this histoplasmosis spore. Etiology means causative agent. So here the causative agent, the fungi which is responsible for this histoplasmosis, is Histoplasma capsulatum. And then morphology, about its structure, the Histoplasma capsulatum is a dimorphic fungi. That means it exhibits in 2 forms, yeast as well as mold form. So mold form, which was which mainly adopt in environment condition, and then yeast form, while it enters into the tissue, it will convert it morphology as a yeast form, okay, round form. And then these fungi grow well in soil as well as artificial media, and then it produces a septate. Okay, it belongs to septate mycelium. That means possess cross wall behind its nucleus. So these septate produce 2 type of asexual spores. First one is a large and round macroconidia, and another one is small elliptical microconidia. Okay, the large macroconidia, its size ranges from 8 to 12 μm in diameter, and then the small conidia, which ranges from 2 to 4 in diameter. Well, and then now we are going to see about the pathogenesis. Here it was otherwise termed as clinical picture. This histoplasmosis is primarily an intracellular disease of reticuloendothelial system. The infection, which mainly targets liver, spleen, hair patches, lymph node, etc. So these overall system together termed as reticuloendothelial system, right? It's an ordinarily asymptomatic as well as relatively mild and self-limiting pulmonary infection. Sometimes it becomes chronic or acute disseminated disease with poor prognosis, right? So their infection mainly takes too long incubation period, and then most infections, 90 to 95%, are asymptomatic. However, due to heavy exposure to spores of the organism, or due to some underlying disease or immunological deficiency or immunosuppressive conditions, these manifestation does occur. And then the lesions commonly produced are of 3 types. So first one is acute pulmonary histoplasmosis. In this condition, it's an acute influenza-like illness characterized by fever and non-productive cough. This condition is self-limiting, as well as on recovery, it produces discrete calcified areas formed around as well as on the lungs. And then second is chronic pulmonary histoplasmosis. It occurs either due to the reactivation or heal of old lesions, or due to the formation of cavities in lungs due to the permanent lesions, right? So mainly some people which already got affected by these primary histoplasmosis, after recovery, okay, that the old lesions sometimes the spores present in the old lesions get reactivated and will leads to this secondary infection. And then third one is disseminated disease. Due to widespread of the involvement of reticuloendothelial system, any organ of the body may involve, including the enlargement of liver, spleen, adrenals, as well as kidney, with associated with fever and malaise. Sometimes the nerves, okay, system, that is the nerve system, may also get involved in these advanced cases. Such infection is mostly found in infants and in immunosuppressive patients such as AIDS patients, HIV patients, in old patients, aged patients, and some malignancy cased patients. And then these are all of the pathogenesis of histoplasmosis. And now we are going to see about the laboratory diagnosis. So the specimen collected for this disease are skin scrapings, pus, bone marrow, sputum, urine, lymph node, and skin biopsy. Direct microscopy. Here the smear made from the pus sputum is stained with Genza or right stain and observed under microscope. While observing under microscope, the organism appeared as a small, round to oval cells ranging from 2 to 3 and 3 to 4 mm in diameter, present within the macrophages or monocytes or in free tissues. Okay, and the culture. The above specimen inoculated in sabrodextrose agar SDA or brain heart infusion agar BHI with chlorophenicol as well as cyclohexamide and incubated at 25 degrees Celsius for 1 week. Okay, why we are adding this chlorophenicol and cyclohexamide means bacteria having 24 hours incubation easily contaminate these fungal plates. Okay, for the inhibition of bacterial growth, these two antibiotics get added in the agar medium. There occurs slow growth. The colonies rise cottony first and which becomes brown color later. So in the microscopic examination of such growth, it shows two type of septate, okay, and which possess two type of conidia. So first one is round and large conidia ranging from 8 to 14 μm, and second one is round to pyriform conidia. This is otherwise termed as elliptical small conidia. Its size ranges from 2 to 4 mm. And other deduction test for histoplasmosis is histoplasmin test. The mycelial phase of the organism in growth culture is filtered, and this culture filter is used for histoplasmin test, which was similar to that of Montauk's or tuber sling test, which is used for the diagnosis of TB. Right? As similar to the Montauk's, here the histoplasmin filtrate gets injected in the patient's forearm, and after incubation period, the induration which predict the patient is positive. Well, and in also old past patients also get answers for these histoplasmin result. Okay, whether they are present or past infection, it indicates the presence of induration, but it does not differentiate the active as well as past infection. And then serological test. So mainly the serological test include complement fixation test, precipitation test, as well as lactose agglutination test. And the treatment is available. The administration of amphotericin B in intravenously mode is the treatment of choice. So thank you for your listening.